this lab, we're going to focus on development and morphogenesis, which are the processes that shape how a plant grows and what it looks like. Instead of just asking how fast a plant grows, we're going to ask how plants form roots, stems, branches, and leaves, and what controls those changes. A big part of that story involves plant growth regulators, also known as plant hormones. You'll be looking at classic experiments that show how these chemicals guide plant form and development. By the end of this lab, these are the skills and ideas you should be comfortable working with. You should be able to describe the major classes of plant growth regulators and explain what they do in plants. You'll also practice explaining how auxin, gibberellins, and ethylene influence growth and development in different tissues. A big focus will be interpreting experimental results, not just memorizing definitions, especially for roots, stems, and buds. You should also be able to distinguish between hormonal control of tropisms, elongation, and senescence, and finally connect what these hormones do in real developmental responses you see in plants. When we talk about development, we're talking about how a plant changes over time, from a seed into a mature organized organism. Morphogenesis specifically refers to how the plant's shape and structure are formed. This isn't just about getting bigger. It's about where roots form, how stems elongate, where branches appear, and how tissues become specialized. These processes depend on coordinated cell division, cell elongation, and differentiation working together. Plants control their growth using plant growth regulators, sometimes called plant hormones. These are chemical messengers that are often made in one part of the plant and transported to another, where they trigger specific responses. Unlike animal hormones, plant hormones don't usually work alone. They interact, overlap, and sometimes even counteract each other. The same hormone can have different effects depending on the tissue and the concentration. There are several major classes of plant growth regulators, but in this lab we're going to focus especially on auxins, gibberellins, and ethylene. Each of these play a different role in plant development. Auxins are strongly involved in cell elongation, root formation, and tropisms. Gibberellins promote stem elongation and can overcome dwarfism. Ethylene is unique because it's a gas, and it plays a big role in fruit ripening, senescence, and abscission. What makes plant hormones so interesting is that they don't just control whether a plant grows, they control how it grows. In this lab, you'll see how hormones influence root formation, stem elongation, bud growth and branching, tropisms like phototropism and gravitropism, and even aging and leaf drop. All of these are examples of development and morphogenesis in action. Instead of running long experiments from scratch, you'll be analyzing results from classic experimental setups. You'll compare treated plants to controls, look at how different hormones change growth patterns, and connect what you see to what each hormone is doing in the plant. The goal is to interpret real biological responses, not just memorize what each hormone does. The big idea to keep in mind is this: plant growth is regulated, not random. Development is directional and controlled, and plant hormones play a central role in shaping form, not just size. By the end of this lab, you should be able to look at growth response and start reasoning about which hormone is involved and why.